Good evening, everyone. I love a good call and response. Thank you all for coming out tonight. My name is Beth, as some of you know already, and I'm new to the See and Learn team. Let me begin by thanking Carib Trans. They are the sponsors of our live stream tonight and every night, so people are tuning in on Facebook as well as here and in person. I should also thank Juliana's, and we've got a couple of other amazing hotel sponsors that we should thank here, including Hummingbird Villas and the Cottage Club for sponsoring our speakers and our visiting presenters this week. None of this would be possible without the support of these amazing local companies, so we are very appreciative of their ongoing support. I should also thank Prince Bernhard Culture Funds and public entity SEBA. They are the primary sponsors of See and Learn, and it's a huge, amazing outreach opportunity, and it wouldn't be possible without them, certainly. And our 50 community sponsors, probably over 50 at this point. So we've got a ton of amazing individuals and organizations that all contribute to make See and Learn happen, and we are very appreciative of their support. For those of you that have not yet seen it, we also have a really new, exciting opportunity to engage with and connect with the culture and the nature of SEBA. This is a soft launch of I'll Explore, which is a mobile platform where we've got really cool interactive stories and photos and information about what makes this island special. So if you haven't yet, you can take a picture, you can scan the QR code, you can go to the platform on either your phone or on your computer and learn and engage. And if you haven't heard yet, we do have raffle tickets that are available for $5 each. So you can buy one, you can buy 5, you can buy 10 or 20 or 50, and you can win really cool, amazing prizes. We've got stays at villas. We've got several different villas here on SEBA where you can win a stay. We've got artwork and jewelry and books and knives and a stay in St. Croix and a trip aboard the Caribbean Explorer and all kinds of really cool prizes. So get some raffle tickets and support See and Learn and win amazing prizes. Coming up later this week, tomorrow afternoon, you can join our presenter tonight, Erin Pilnick, for a really cool opportunity to go out and explore the local diadema dive. Oh, it's full. If you haven't signed up, too bad. You're missing out. I'm going and it's going to be fun. Then Tuesday night, we've got another presentation at Liam's Cuisine on Animals That Ink with Lauren Simonitis, who's here in the audience as well. Hi, Lauren. We're very excited to have you here. And then on Wednesday, you can join her for an afternoon dive that's going to focus on inking animals as well. So come by and visit us at the tent and sign up. If you sign up and attend a field activity like one of those dives, you get a very cool See and Learn shirt, or we also have ladies' tank tops, and they're all made out of recycled plastic, and they have additionally been sponsored by the Lions Club, so huge thanks to them for that. And who's got one in their hands? Very nice. Yes, we do recommend you try, if you haven't yet, SEBA Cloud Top. Feel on top of the world with SEBA Cloud Top that has been brought to us by some amazing friends and partners and former and current experts as part of See and Learn. So it's a great beer inspired by SEBA Spice. So on to tonight's keynote speaker, Erin Pilnick. Erin is actually another colleague of Alwyn's who's here to present another win in restoration efforts. So this time he'll be focusing on corals. Erin earned a PhD in interdisciplinary ecology with the University of Florida in December 2022. His main research interests include restoration aquaculture and coral reef ecology, with an emphasis on marine invertebrate biology. Beginning in 2018, Erin has also worked in close partnership with the Florida Aquarium in Apollo Beach, Florida, to improve aquaculture and restocking methods for an ecologically important reef herbivore, the long-spined sea urchin, or Diadema antillarum. The development of a novel recirculating aquaculture system has led to the refinement of larviculture protocols, the production of over 1,000 juveniles, and subsequent first attempts at restocking hatchery-reared urchins into the wild. So for those of you that are signed up and are lucky enough to join us, you can come out with Erin to see restoration in action. But for now, sit back and enjoy his presentation. Let's give it up for Erin, everyone.
Cool. Thank you, Beth, for the introduction. Thank you to the See and Learn program for hosting this. It's really an honor to be here, and also probably the most beautiful place I'll ever give a talk in my life. So it's going to be a big one to live up to. And also, I just want to say thank you to our Dutch colleagues, Alan and company, for their hospitality throughout this whole trip, and it's really just wonderful to be here with you guys. Cool. So yeah, tonight I want to talk about some of the land-based coral restoration work that we do back in the United States, and I think it's interesting because we're about 500 or 600 kilometers away from a coral reef where we do our research, but we're innovating some new technologies that I'll talk about in a few minutes. That will hopefully advance coral restoration in other areas of the Caribbean and the world. So cool, we'll just get right into that then. So unfortunately, and it feels kind of wrong to talk about, to start with the doom and the gloom in such a beautiful setting, but I kind of have to just get this stuff out of the way. And why are we even talking about coral restoration in the first place? And so this might not be new information to people, but essentially, big picture, we've observed this global collapse of marine species since the 1950s. And then looking even further back in time, we can start to attribute some of these losses of different types of groups and ecosystems to human influence. And so we've started to see the large declines in these groups and these systems around the same time that of the emergence of some global markets. So things like coastal land development and eutrophication and excess nutrient pollution has led to the declines of a lot of these systems. And for those of us who are studying coral reefs in the Caribbean, the story's much the same. So this graph gets shared a lot, pretty frequently. But in this one, it's the data that's reported here is the percent coral cover over time. So since the mid to late 1970s, we've seen about a 40 to 50%, or even greater in some regions, reduction in the percent of coral cover in these regions. And so given that, our notion of what a healthy coral reef is and what it looks like has really changed over time. And so I like to include this slide just to show, some of the dive operators in the Florida Keys are basically providing these highly photoshopped images of really colorful, bright reef fish, but in seagrass habitat to try to encourage diving in the Florida Keys. And next one's my personal favorite, photoshopped images of Indo-Pacific fish and corals that we don't even see in the Caribbean as a way to promote diving in the Keys. And so it's really interesting because this is really what we have observed is this decline in coral reefs over time. So, in the top picture on the left, this is from Carey's Fort Reef in the Florida Keys. And this is kind of our prototypical, really healthy, beautiful, vibrant reef with a high percent coral cover of elkhorn coral there, a high degree of structural complexity, lots of habitat for corals and fish, and really vibrant. And then over time, we've seen this degradation of the habitat and of the ecosystem due to a number of reasons. And what we see now is this real, basically, in some of these reefs, we don't really see any live coral anymore. And what's replaced a lot of the live coral is a lot of macroalgae, which I'll talk about in a little bit as well. But we've seen this massive reduction in structural complexity, and that's a really big, really important thing to discuss when we're talking about coral reefs. So, but despite this collapse, we're still seeing a lot of usage of these coral reefs. So here's some interesting data from 2019, and this is reporting some of the visitation statistics within the Florida Keys National Marine Sanctuary. So from 2019, we saw that there was about 1.86 million people who visited the marine park and participated in snorkeling and diving, and this attributed to about $1.2 billion in industry value. And then also we like to talk about natural infrastructure of reefs. So a healthy coral reef is this biogenic ecosystem that's continually calcifying and growing, and it provides a lot of coastal shoreline protection. And so there was a really interesting study done in partnership with Lloyd's Insurance Group. I'm sure many people have heard of them, that tried to evaluate what coral reefs are worth to the U.S. Coastline, and we think that it provides about $1.8 billion in annual flood protection benefits, and that's just per year. So when we talk about natural infrastructure, healthy coral reef really does provide us an immense amount of value. So okay, enough doom and gloom. Now I kind of want to transition and talk a little bit about some optimism. So despite these trends, we're as a culture starting to realize the importance and the value, the economic value of these systems, and so we're starting to be able to reverse some of these trends, which is really cool. This is really good, promising and optimistic data. So we've seen some trends like the recovery of seagrass meadows in some in parts of Europe. We've seen some sea turtle conservation success stories, and also humpback whale populations have been recovering in off the coast of Australia. So, there's a chance here that we're starting to do the right thing. And so reef recovery is also possible to do. And so here's an example. This was a study that was published in 2002, and they looked at the coral assemblage 50 years after a chronic localized anthropogenic disturbance, and which you might realize was the detonation of about 70 megatons of nuclear weapons. And so this is pretty cool, where we saw 70% coral recovery after this localized disturbance event, which seems to be underplaying it in my opinion. But so for Bikini Atoll, we think that, so ironically, after all this nuclear testing, we sort of kept humans away from developing and from polluting those ecosystems. So that's maybe one of the reasons why we think that we saw that recovery. And then another factor is that Bikini Atoll is relatively close to other coral reefs. So if there are upstream reefs that are providing larvae, corals can basically move from atoll to atoll. And so the fact that it was close to another coral reef really helped out with some of that recovery. But returning back to the Caribbean, we really haven't seen this recovery. And so there's a number of, it's sort of a complex question about why. But we think, there's this basically, if a coral reef is subjected to one disturbance or one stressor event, even a nuclear explosion or multiple nuclear explosions over time, it's fairly resilient and it can bounce back and recover from that. But when you have these compounding stressor events, we started with in the Caribbean, there's human influence of nutrient pollution, and some disease that started the decline and started that ball sort of rolling down the hill towards a degraded reef. And then stressors that have compounded the problem, overfishing and the loss of herbivores, and I'll talk about that in a little bit, as well as the more recent problem, which is climate change and bleaching that's associated with ocean warming. And so that's really led us to where we're at now. We're in this sort of stable ecological state of low percent coral cover and high percent macroalgae. And so the point I want to make is that coral reef recovery is possible, but difficult if we keep compounding these stressors on top of each other, and it sort of becomes more of an uphill battle over time if we don't start to address all of these stressors. And so for global warming, and ocean warming, this is really the Achilles heel for coral reefs. So coral reefs have a really narrow thermal tolerance, and when the water temperature starts to rise, then that's a really large issue. So even in the event of a warming to 1.5 degrees Celsius above pre-industrial temperatures, we're still expecting about a 70 to 90% loss globally of coral reefs. And we're probably going to exceed that temperature threshold. So we have to think about a number of recovery actions that can all work together to try and attempt to manage this problem. And so I think the big one is really to tackle climate change and start to mitigate greenhouse gas emissions, which we all I think we all see that pretty clearly. But some other recovery actions include pollution control. right? We need to start to manage the amount of nutrients that we're putting out into the ocean because these are really negatively affecting these systems. Expansion of marine protected areas. These are all really, really important things to start to try and do and to do more of. And so one sort of sliver of this recovery action, so because we're losing reefs so rapidly, the rate of decline is so rapid, we can also start to think about some active restoration measures as an additional component, as an additional recovery action, something that we can do, but we need to do it in conjunction with all of these other things that we need to start to address. So, okay, so what does coral restoration really even look like? And I'm sure, again, a lot of people here are really familiar with this, but the general strategy that has been in place and has been utilized for a number of years now is this concept of coral gardening. And this is from a picture from the Coral Restoration Foundation, which is located in Key Largo in the Florida Keys. And the general principle is this is kind of like an agricultural model, but for coral restoration. So we'll take these fragments of corals, hang them on these nurseries in these coral trees, and then that's a way to produce this biomass, right? So we can use that; they can grow on the trees really effectively, and then those corals will get fragmented and then brought out to degraded reefs and outplanted. So that's great. This is a strategy, but unfortunately, also over time, what we've seen is that the long-term success or long-term outcomes of this restocking is not so great. So after only a couple of years, we see that the survival goes down pretty far, and this is because of all of these stressors that we're talking about, and also. As we saw recently with this past marine heat event, a lot of the nursery stock has bleached out and has actually died from the Florida Keys. So in general, this is restoration is sort of a high risk, high reward strategy, I would say, and it's high risk because we could try to grow a lot of this biomass in nurseries in the ocean, but then if we have a particularly warm summer and it exceeds their temperature threshold, then it's a lot of work for nothing essentially. And so that's kind of where we're at as in the restoration community is we're trying, we're just sort of coming out of this bleaching event, especially in the Keys where it hit us and where we do our research particularly hard. So we're trying to sort of grapple with what comes next and how do we continue to approach the problem. So now I can kind of just talk a little bit about the biology of the restoration, and then we'll get into sort of the reproductive biology. But a lot of what's going on in this restoration work in coral gardening uses a technique called fragmentation, and this is essentially asexual propagation. So corals, they are animals, but we can take a piece of coral and snip it off from a parent colony and then move it onto a tree, and then it produces biomass. But the issue, or one of the limitations of the strategy, is that these fragments are genetically identical to their parent colonies. And so really in these nurseries, if we're doing all this asexual propagation, we're maintaining low genetic diversity populations. And that can be a really big problem if we know that these individuals are susceptible to disease or they don't have high thermal tolerance. Then we're essentially just propagating those same genetics and putting them out to the reef and seeing the same problem occur over and over again. And so there's really no room for adaptation of these corals if we're doing this asexual propagation. So thankfully, corals can have sex, which is cool. So corals can sexually reproduce, and we can go through this process of sexual propagation instead of asexual propagation. So this is a little bit of the reproductive biology here. So most of the corals that are hermatypic, or what we call these reef-building corals, are actually broadcast spawners. So that means that sometimes we'll have gonochoristic or male and female individuals, with males releasing sperm and females releasing eggs into the water column. And then there's a process of external fertilization, and then the embryos go through embryogenesis and develop into larvae. The larvae are actually, this is really interesting, I think. They're modal planktonic larvae. So they're able to swim around the ocean until they can find a habitat or a piece of reef that they want to settle onto. And then they'll do that. And then once they do that, they become sessile. So they will settle and start to calcify, attach, and then they'll become a living coral right there. And the benefits of sexual reproduction is that we can really, this is a way to generate novel genetics. And if we can have higher genetic diversity in our populations, there's a chance that some of these novel genotypes could have resistance to disease or have higher thermal tolerance, et cetera. And it's the same thing like when we're talking about agricultural models and why we want to try to avoid monoculture crops because there's a high risk of disease wiping out a monoculture. So, okay, so we know that corals can sexually reproduce, which is really cool. Now I want to talk a little bit about how they go about doing that. So a lot of these corals will have they're a highly synchronized annual broadcast spawning event. And so essentially, you'll have members of the same species of different corals or colonies that will cue into really, really specific environmental mechanisms so that you could have multiple individuals spawning at the same time on a reef, and often in really remote, they could be really remote regionally from each other. And so it's a really interesting, we think it's an adaptive strategy to maximize some of the fertilization success. So imagine it's a really big ocean and you're expelling gametes into the ocean and they're so dilute that you need to have all of your individuals spawning at the same time so that the gametes can actually meet in the water column and fertilize and then become corals. But for this process, the timing is really everything. It's critical because if there's a mismatch in timing and they don't spawn at the same time, then there's a low chance of that fertilization occurring. And so we think that there's a number of environmental mechanisms that act on the corals to basically cue them into spawning at these really specific time intervals. And it happens at different time scales too. So we think corals may know what season it is generally from water temperature or changes in water temperature. Similarly, they might know roughly what month it is or the relative month due to the photoperiod or the amount of daylight that the corals are subjected to. And then getting even more refined down, they sort of know what time of day it is, or what day it is, sorry, based on these lunar cycles. And then even further, they might know what hour of the day it is based on how much exposure or absence of light there has been after sunset. And so they use a combination of all of these environmental mechanisms to really control, or they're sort of programmed to all spawn at these really, really specific time points. And so this is a figure just to kind of drive home the point that so over time, coral biologists have done a lot of really interesting work to try to be able to predict and understand when these corals are going to be able to spawn. Right. So for the coral on top, that's the grooved brain coral. Just for example, we think that in Curacao, because this is from the Carmabi research station, that they will spawn 11 to 12 days after the full moon, which would correspond to the 18th of May, the 16th or 17th of May at 5:42 to 7 p.m. And this is really how specific the timing of the spawn is. And I think it's really incredible. So the biology underlying the mechanisms for how they go about spawning is really, really interesting. So, okay, again, why are we even talking about coral restoration? So if there's this natural reproductive process, why do we need to step in? And the problem is that the natural reproductive success is really limited. So we have low population densities. We've lost a lot of these corals over time. And so while they may be spawning at roughly the same time, there's just not enough individuals that are producing gametes to produce these reproductive populations. And also the habitat has changed over time. So now I mentioned earlier that we have more macroalgae on our reefs and less structural complexity. So even if the fertilization does happen and the larvae are swimming around, sometimes they can't find a piece of reef to settle to and then go through this metamorphosis and settle and become a juvenile coral. And so really what we're thinking, where our mindset is right now is that so these stressors and these effects of climate change are really outpacing the rate of natural adaptation. So they're not reproducing fast enough, or there's not enough sexual reproduction that's happening in order to have enough inherent genetic diversity to be able to deal with some of these stressors that are occurring over time. And so that's, again, where we step into this process. So whereas we were talking about asexual propagation before, now we can try to do some sexual propagation of corals. And what this really involves is night diving during those specific time intervals where we think they're going to spawn, and then we'll put some nets over these corals, and then hopefully they'll spawn. If they do spawn, then some of the gametes will be collected, like there's a collection cup up there, and then the gametes can be brought back to the lab, and then the larvae or the embryos will be fertilized, and then develop into planula larvae, and then we can put them into these environments, in these tanks, essentially, or these small mesocosms, where we then give them an opportunity to try and find an appropriate substrate to settle. So in the video on the left, these are some planula larvae. So now this is a couple days after successful fertilization, and they've developed into larvae, and then they're basically swimming around in that tank and trying to find a good place to settle. And then in the top right, different species of coral, but that's what the recruit would look like maybe a couple months after they settle onto that environment. So now we're talking about this process of sexual propagation. Logistically, very, very difficult. So you can imagine it takes lots of people, lots of boat captains, lots of time on the water, huge teams of people, night diving. If you're out at 7 p.m. and you get a spawn, and then they're spawning for 2 hours, then you come back to the lab, and then you have to go. Sometimes you're working through the night to get these corals for the fertilization and for the larval development. And so logistically, it's very difficult. And sometimes if these corals are spawning once a year, and if you have a storm that's rolling through at that moment, then that's sort of it. You've lost your chance to collect gametes for the year. Right, so it's difficult. And this is compounded even further by recent disease issues that we've been seeing. So even if we do go out into the field, which we still do, there's just fewer live reproductive colonies out there to collect gametes from. And so this is 2 figures of stony coral tissue loss disease, which I know you've had guests talk about this before. I think Aaron Muller last year gave a great talk on stony coral tissue loss disease. But it hit us really hard in 2019, especially in the Florida Reef Tract. And we started to lose a lot of these reef-building corals. And so there's just fewer out there for us to be able to dive and collect gametes from during their natural spawning events. And I just wanted to include this slide just to kind of show how the dire state of this disease progression. So it's an infectious disease that causes tissue necrosis. And within about a month to 2 months, you could go from having a really healthy coral all the way on the left there to complete mortality within that time period. And this is sort of a progressive infectious disease. And so that kind of leads nicely into the research that we're doing. So as I mentioned earlier, we're based at this, it's a land-based facility. So it's really nice to come visit Saba and be able to dive right here and to visit Alwyn's lab, because you can just jump in the water and design your experiments from lab to reef. But for us, because we're hundreds of kilometers away, if we want to go try to test some of this work in the field, it requires a lot of logistics. That being said, there are a lot of benefits to having this land-based facility, which I'll talk about. But this is kind of just, I wanted to give an overview of or a setting of where the research is taking place and where the facility is located. So that's an overview picture on the left. And then on the right, encircled there is Tampa, Florida. So we work on the west coast of Florida. And then a lot of our research is down in the Florida Keys, which is sort of on the southern tip of Florida there. But this is a really interesting collaborative environment where we have this facility. So it's actually owned. The land is owned by an electric company, which is slightly ironic, but I'll leave that out. And so they lease and sublease some of the land to the Florida Aquarium, and then some of that land is also leased to, we have a Florida Fish and Wildlife Conservation Commission. And then we're affiliated with the university, and we're kind of like squatters on the facility. So we're like a satellite of a satellite campus, but we provide some of like the academic or research background for the site. And this is an image of a coral greenhouse. So these coral greenhouses are owned and operated and maintained by the Florida Aquarium. And within the greenhouses, there's a number of these really advanced recirculating aquaculture systems. And so when we think about just conventional aquaculture, we think about salmon farming and these really intensive food production systems. But a lot of that technology can be applied sort of to this work. And so we're utilizing some advanced aquarium technology essentially to house corals and to go through this develop a managed breeding program for corals. And I think you know, as us as academics, we're you know, good at writing papers and proposals, but we're maybe not so good at like building large infrastructure projects like this. And so it's a good, really amazing partnership to be able to work with a public aquarium that has the expertise and the ability to install and build some of this infrastructure to try to attack some of these problems. So this is the Florida Aquarium in one of their greenhouses. Picture on the left there, these are both sexually propagated species of corals. That's Acropora cervicornis on the left and Dendrogyra cylindris on the right, or the pillar coral. And so kind of one of the impetus for developing this facility in part was because we were losing so many corals due to stony coral tissue loss disease, that we wanted to just start taking corals physically out of the ocean and bringing it to a facility where we could maintain them on closed systems, meaning that they're not connected to the ocean water, where you could recirculate the pathogen through the systems, and then sort of generate or develop like a living gene bank of sorts. It's kind of like a seed bank, but for living corals. And so now at the facility, we have 16 native stony coral species native to Florida, 4 of which are listed under the Endangered Species Act as threatened. And those 4 species are labeled down at the bottom there. But wanting to take this one step further, having them as a living gene bank is great, because number one, it just preserves those genetics that we would have lost in the wild otherwise. But we are asking the question: is it possible to develop a managed breeding program similar to other public zoos and aquariums for other endangered species? Is it possible to do this? But it's a really big question, because number one, keeping corals alive in fish tanks is really difficult, because they require really, really clean water. It's certain types of circulation. The lighting has to be very specific, and it's quite difficult to do that. But then on top of that, we're asking the question, well, can we get them to produce gametes in an aquarium? And further, can we get them to spawn in an aquarium at reliable or predictable times, as they might do in nature? And so some of the challenges to start to manipulate some of these environmental mechanisms, but in an aquarium, in a fish tank, such as the photoperiod, or the length of the daylight, the sea surface temperature that changes seasonally, the amount of insolation, or the amount of solar radiation that is exposed to the corals at any given time during the year, further the lunar cycle. And so trying to trick the corals using these environmental parameters into thinking that they're actually on the coral reef, so that they'll go through this reproductive process. And a lot of this work was actually pioneered by Jamie Craggs out of the UK, who was the first to do this and develop a system. And he's located at the Horniman Museum in the UK. And he basically designed and built this system. And I don't want to go through all of sort of the components of it on the left, but I want to drive home the point that there's all of this technology that's involved, which is really sort of new, right? And 20, 30 years ago, we didn't really even know how to keep some of these hard corals alive in aquariums. And now the technology and the knowledge has developed to the point where not only can we do that, but we can start to try to promote reproduction in these systems. And part of the technology is that we have better microprocessors and commercial programming units where we can really replicate. So the graph on the top left is the seasonal temperature from the Florida Keys. So we can program on a monthly variation the amount and change of temperature that occurs over time. And similarly, some of these controllers can actually, we can dial in the intensity of LED lights, so we can provide them a different amount of insolation or solar radiation over time, over the course of a year. And again, I'm not even going to go into any of this stuff, but another point that I want to say is that some of the newer technology, we're really able to get a good grasp on the water quality. And so we're able to send water quality samples off to labs that are able to basically tell us some of these trace elements, concentrations of, for example, copper, which is toxic for marine invertebrates, down to the level of a couple parts per billion in the water. And this has really given us a greater ability to try to track water quality and to manage water quality in these systems over time. And then, fast forward this is basically a picture of what one of these Project Coral tanks looks like. And then, so there's a number of pillar coral colonies in there. This species in particular is quite precious to people at the facility because it's actually, we think, functionally extinct in Florida. So there's just so few individual colonies left that basically the chance of them reproducing on their own is zero, essentially. So we're expecting total extinction of the species within our region within the next decade or two. And so a lot of the corals that are these colonies that are pictured here are genotypes that no longer really exist in the wild. And so we're able to go ahead and spawn these corals here. Another thing that you'll notice about the picture here is that there's all these blackout shades around the aquarium. And this is intentional. So when I mentioned earlier that with some of the field work, we have to go out really late at night and to do the diving, well, with these systems, if we can black out the aquariums, we can actually put them on a reverse photo period, which is really nice. So now we can just basically wake up, have our cup of coffee, roll into the lab, collect some coral spawn, go about our day, and then we can go home and have normal dinner at night with our families. And so this is really nice. So cool. Now I get to show you just some cool videos, some high definition videos of this is the maize coral that was spawning in one of those propagation systems. So that's a male. So this is a gonochoristic species. So that's a male that's actually releasing sperm. And you can imagine also for some of these species that are gonochoristic male and females, if they're releasing sperm in the wild, how are we going to collect that in the ocean, right? But if we have it in a contained environment in an enclosure, we can actually collect a high concentration of that and then go through this fertilization process and produce viable fertilized gametes. And then here's this next video is of a pillar coral. So this is the coral that I mentioned was functionally extinct throughout Florida. And this is a female that's releasing eggs. And again, it's really obvious to see when you've got a close-up, a really nice lens with a camera on it. But imagine if you're in the ocean diving at night, it might be really hard. It is really hard to just see this happen. So yeah, we get a nice view and produce some nice media from this project as well. And then so moving down along the line, skipping a whole lot of work that's involved in like the larval propagation. So on the left is the juvenile pillar corals, and they're also mostly photosynthetic, but they receive some heterotrophic feeding as well. So in the video, I think we're feeding them some krill or some type of crustacean, which is also really important part of the process to make sure that if we're going to keep these corals, these adult corals, these are not of a reproductive age yet, but we feed the adults as well, the broodstock, and we think that nutrition is a big component of how to keep them in sort of a good reproductive condition, so they are producing these gametes in the aquariums for us. And then a picture on the right, these are staghorn corals. This is one of the two really important reef-building corals in the Caribbean. And now the Florida Aquarium basically has sexual propagation of these corals down to a science. And so it's a matter of, at this point, scale and sort of how big can you make a facility, how many people can you hire. It sort of becomes an economics question about how can we start to scale this up as needed to approach restoration. But I think I'll pivot a little bit here to start to talk about some of the dynamics of growing corals in fish tanks. As we're growing these juvenile corals, imagine that you start out with a cell that becomes fertilized, and then when they're actually settling onto these coral tiles, they're microscopic still, and there is a lot of competition for the habitat at that life stage. So you imagine that alongside these corals, you have a bunch of algae that's growing in fish tanks. And this is, if anybody's ever kept a fish tank here, we know that we're constantly fighting algae, right? And so the same thing, there's the same dynamic for growing corals, where if the algae grows too much, it can essentially outcompete or overgrow these recruits, and it can lead to them dying before we can get them up to a size where we can actually move them out to the reef. And so part of the research that we've done and what we're looking at is this using herbivores, small herbivores, and placing them in the aquariums with these sexually recruited corals at the same time to limit the algae in order to improve the growth and survival of corals. And again, this is sort of an economics question because when we don't have those herbivores, it basically takes a team of biologists and interns with dental tools under a microscope trying to scrape the algae away from the coral. And if we can have a snail or an urchin species that does that for us, then all of a sudden your capacity, your production capacity can really increase. And so we've done a lot of work trying to figure out what species of herbivores, snails or urchins, what size of these different types of urchins or these herbivores leads to the best outcomes for growing corals. And it's not always a slam dunk. There are some species that will actually eat the corals as well, and we don't really want that to happen. But okay, so here's just a figure from a publication that we released. So on the top two rows are pictures of corals starting from, on the left, day 1 through day 126 days, and that's coral growth. But those corals were placed in the tank with 2 different snail species versus on the bottom, those corals were, no herbivores were essentially placed in the tanks with those. And what we found was that after time, this same species of coral, when they didn't have herbivory, the survival and the growth was much lower. So some really interesting results there. And then, kind of continuing this thought process about algae and coral interactions and dynamics. We have the same ecological dynamic that is happening on the reef between corals and algae. So I think you've probably heard some talks about diadema in See and Learn before, and this is actually sort of more of the focus of my dissertation when I was in the PhD program. But essentially, for Caribbean coral reefs, we have had this loss of herbivores in the mid-1980s, in part because of overfishing of parrotfish, and then also in part because of this mortality event that affected diadema, these long-spined sea urchins. And with that, like in an aquarium, where if we don't have the herbivores, the algae starts to overgrow, and it really degrades the habitat to the point where corals can't recruit to the reef, and they're actually overgrowing some of these coral outplants. And then, so this is sort of outlining the process of the ciliatosis, which I think Ian Hewson talked about this year in the program. So I won't get too much sort of into the disease dynamics, but one of the other projects at the facility that we work at in Tampa is trying to understand and improve diadema aquaculture. So again, this is aquaculture technology, not for food fish production, but for herbivores that then would be restocked to the reef in order to provide some of that herbivory that's been lost over time to then basically improve the habitat for the recovery and restoration of coral species. And so this is a graphic just showing basically how the diadema populations were affected by that initial mortality event in 1983 and 1984. And in most areas we haven't really seen a dramatic recovery, a natural population recovery of these herbivores. And so now we're in this stable state where coral reefs are really overgrown with macroalgae, and it just really makes it harder. It's one of those stressors that is making it harder for us to fight that uphill battle to recover some of these coral reefs. And there's a photo from Alwyn in the top right from the last mortality event in 2022. So here's a conceptual diagram of sort of what that process would look like. So really similar with the corals, we would take wild urchins into a land-based facility and then maintain the brood stocks so that they're becoming reproductive and they're producing gametes, and then induce them to spawn and then go through this larval rearing process, which is arguably more difficult than getting corals to spawn. Because the larvae are incredibly, I'll talk about that in a second, but they have to grow as planktonic larvae for a period of 30 to sometimes 90 days long, where we're feeding them microalgae and trying to understand some of the nutritional requirements in order to get them to settle and then get them grown out big enough where we can actually start putting them back out into the wild. So culturing diadema is also really, really, really hard. It's incredibly labor intensive. We don't have a commercial process, so a lot of what we're doing at the facility is R&D, research and development. And as I mentioned, the larvae are really difficult to grow. They're incredibly sensitive to heavy metals. So they're probably what we think is the most sensitive marine species tested for dissolved nickel and selenium. So going back to the water quality graph earlier, we're constantly checking for some of those parameters to try to understand what is causing, how to get them through the larval development period. They have really specific nutritional requirements, and then even in aquariums, we're dealing with disease issues like corals are in the wild. So they're just incredibly sensitive to pathogens. And so here's what a larva looks like, sort of a healthy larva in the top right, and then unhealthy in the bottom. And so this took many years of sort of trial and error in trying to understand what are the developmental impediments and how do we get these things to actually grow in fish tanks so that we can get them grown up and out to the reef. So similar process, broodstock urchins are collected in the Florida Keys and transported up to our lab in Tampa. We constantly maintain a broodstock population on artificial diet and then induce them to spawn, extract the gametes, and then move them up into an experimental recirculating system. So again, applying some of the same technology that we did with the coral repro tanks. And also trying to build in enough tanks so that we can really experimentally look at how this process works. And actually, interestingly, if anyone's been to an aquarium and have seen these jellyfish tanks, we call them chrysals, and they're really designed to keep the jellyfish really gently suspended. And so if it's too harsh, they'll get ripped up on the sides of the aquariums. And we applied a similar design to growing urchin larvae here. And so we adapted some of these tanks into the larviculture system. This is always a fun photo. I was really sort of excited and happy in the before picture, and then maybe slightly less happy afterwards because there's a lot of blood, sweat, and tears in building the lab. But along the way, we've done some research looking at different microalgae diets. So again, nutrition is a big thing for corals and sea urchins, and like in any aquaculture process. So we're trying to find an appropriate microalgae that has the right amount of protein or the types of polyunsaturated fatty acids. A lot of things that apply to human nutrition as well. And then here's a cool picture of basically after a couple of years, we completed the life cycle, and now we're able to successfully, after all of that R&D, pretty reliably produce somewhat high numbers of these urchins. And now actually we've produced I think over 3,000 of them. So we're getting better and better and better at that, and trying to fold that into the overall restoration strategy. So here's a cool video. Hopefully this will play. Yep, this was produced by the Florida Aquarium, and I'll kind of annotate over the process. So that's one of our broodstock. that's removed from the system. Here they're getting placed into an iodine bath for disinfection, and then they're placed into this container where we increase the temperature, and then there's a female that's spawning, and so that's producing eggs there. And then we'll extract the gametes really carefully as we can. And there's our team, and then we bring them over to some bins, and then we'll retain the eggs, but start to flush the sperm out of the container to prevent polyspermy or water quality issues. And then they're loaded into these chrysals that I had just mentioned. A lot of times we're just looking at gametes under a microscope. And then, again, we skipped about like 2 months worth of really hard work, and then we just end up with sea urchins. So that part's really cool. And so, yeah, some of the research along the way where we've looked at different we're able to take a real deep dive into the larval development and the larval biology, trying to understand what does the morphology look like. And so in a way, this is really applied research because we're really trying to produce a technology for restoration, but we're able to learn about the basic biology of these animals at the same time. Things like what do they settle onto? So the same thing with these planktonic larvae floating around the ocean, and then if they encounter a reef, what are some of they have certain settlement cues that they'll like to see and then they'll settle onto those cues, and then undergo this process of metamorphosis and settlement into a sea urchin. So we had a question, well, what is that? Because this is really hard to try to understand in the field. Because they're these microscopic larvae. How are we going to collect them from the wild, and how are we going to, really do a really controlled experiment looking at some of these different parameters? And so, by us being able to aquaculture them in the lab, we're also able to learn about some of the biology and the ecology of the animals. And I just wanted to include this also. So we've worked in real close collaboration with Alwyn. And also Tom Weigers, who visited us, I think in 2021, was it? And so it's been a really fruitful collaboration with them over that time period. And we've worked together on trying to advance this technology and the research. And they've come up with some really novel ways to grow diadema sea urchins as well. And so, and then we've been able to apply some of that to some of the processes in our lab. So, yeah, really thankful for the collaboration and hoping to be able to continue some of this work with them into the future. So, I just kind of want to end on what are some actionable items of restoration. This is from a paper we published in 2021, where we took some of the sexually propagated corals. And we were, interested in. We had some questions like, of what age do they need to be, or at what are there different survival outcomes depending on the age of the corals. So are there different cohorts that we can put out at different times to improve the survival, and also are there different outplanting methods, whether they're sort of epoxied onto a tile and then onto the reef, or just nailed right down into the reef. Are some of these methods better or worse for actually getting corals to survive once they're outplanted onto the reef? Also, we've done the first legal or permitted release of hatchery cultured urchins, and I just want to make sure that people know it was permitted, of diadema sea urchins to some Florida reefs. And so we're still trying to understand, this is sort of the next line of the research, is to try to understand, okay, well, now we can grow them and in somewhat large numbers, but what does the process look like for actually putting them back out onto the reef and getting them to eat algae for us? And so, yeah, I'll conclude by, I think the talk was really sort of trying to focus on sort of the type of work that could occur and happen at a land-based facility, even hundreds of kilometers away from a coral reef. And just to recap some of that, it's basically the facility is acting as a living gene bank. So even if none of this restoration really is fruitful and sort of we lose all the coral reefs, which is sad, but at the same time we'll hopefully be able to just preserve some of these genetic lineages at this facility over time. And then the technology also gives us the ability to do some assisted evolution, where we can start to look at and select for certain traits, such as disease resistance or thermal tolerance, And as mentioned, we can just increase our biological understanding of these animals in captivity. And that's it, really. I just want to, despite all this, I really want to sort of hammer home that while we are working on this, and it seems really optimistic, we still have a lot of work to do, right? So, this one component of rest, recovery is not really going to get us all the way there. But I want to leave people with sort of an optimistic feeling about that we are doing some things to try to tackle the problem, and we just need everyone else's help to kind of get on board and get some more work done. So with that, that's it, and I'd love to take any questions. We have had a number of collaborators and funders, and too many to name here, but they're on the slide here. Yeah, and I don't know if I think I have some time for questions. Yeah.
Yeah. Hi, thanks very much for your talk.
Yeah.
I've got a question about the videos showing the gamete release by the coral. Two-part question. Number one, was the video in real time? Second part, that seems very highly synchronized. Is that just down to the environmental factors you talked about, or is there some kind of chemical signaling within the colony?
Yeah, great questions. Thanks. So the video was it was in real time, so it wasn't sped up or anything, and so that was the speed of the gamete release. And to answer your other question, we think mostly it was due to manipulating those environmental parameters that I talked about, but there are probably things acting on their spawning biology that we don't understand. For instance, we suspect that if there's gonochoristic corals, one male and female, that maybe the sperm, if the female colony encounters that, that might induce the female colony to release eggs. And so there might be some sort of chemical interactions there, but I don't think we haven't really fully flushed that out yet.
Okay, thank you.
Yeah, thanks. Yeah.
How long after release are the coral gametes able to fertilize?
Also, a really good question, and I think it can be species specific. So some, I think it would really, the limiting factor is probably the sperm motility. And after a while, if they don't encounter the egg, there's probably, I think maybe an hour or two. Yeah, within a couple hours, and I think that could change depending on species, but basically however long the sperm is viable for. Yeah. Yes?
The word gamete has been used numerous times. What does it mean?
That's Thank you for that. It's a reproductive cell. Oh, sorry. Yeah. What is the word? What is a gamete? Thanks. It's a reproductive cell. Sperm and egg. Sperm and egg cell. Yeah. Yeah.
Hey, it was a great talk.
Thank you
so
thank you.
much. So, kind of in the lines of what was asked earlier, is any work being done on, instead of like controlling the parameters, like looking at the receptors themselves, like the photoreceptors, the temperature receptors of these animals, and manipulating that, like genetically?
So the question is, in addition to the environmental parameters, are we looking at any cellular biology, like the receptors, and trying to potentially manipulate that to induce spawning? Is that right?
Well, just like just not controlling the outside parameters, but how they are receiving the parameters. So how they are detecting that difference in light or temperature.
Yeah.
At the animal side rather than the external.
Right. So trying to manipulate the cellular biology instead of the environmental parameters. So also a good question. I don't really know. I think there's probably some research into that. And yeah, thank you for that question, because now I need to go and do some more reading. Yeah. Yes.
Are you using the corals that you have? Are you using just ones that you got from around where you are, around Florida, or do you get them from further away as well, like Curacao, where you have?
Right. And the question is, the corals that we have in the managed breeding program, are they mostly from the Florida region, or are they from other regions? So most of them are from Florida, and so they were kind of collected from the Florida reef tract ahead of the disease progression. But that being said, there have been at least one project where we've actually transferred like gametes from Curacao or Bonaire and mixed them with gametes from Florida populations. So I don't think we don't have individual colonies from those other regions that are crossing over to different areas yet, but we're able to transport the eggs and the sperm in order to do some targeted crosses. Mm. Yes.
What's the medium that you use to grow the staghorn coral on? It looked like pieces of styrofoam.
Right. So the question was, what is the substrate or the medium that we're growing the corals on? And great question. Sorry, I didn't mention this. They're ceramic tiles.
It's quite thick, though, that it showed the staghorn coral.
Okay. Yeah. So the I think you're talking about this image here. This one and this one, right? In the top left. Is that what you're looking at?
No, it's before that.
Oh, okay. So the ceramic tiles that where we get the coral larva to settle on in the lab, we can basically design how thick those are. But most often they're not really that thick. They're only a couple centimeters.
Yeah, it looked like a piece of styrofoam, the thickness.
Okay, I'm not sure. We use a number of materials. So maybe you're thinking about we use egg crate. It's kind of like it's a lighting material that you might see like in a. Let me see if I can find the photo. I'm not sure. I might have to find it later and then I'll come talk to you. Okay. Yeah, but we basically have them settle onto ceramic tiles.
Okay.
It has to be kind of a hard substrate that we will leave in the systems and allow them to kind of grow the algae and other substrates onto the tile that the corals will then queue into in order to settle.
Okay, thanks.
Yes.
What are you doing to condition the lab-grown diadema to cope with currents and predators and things like that?
Yeah, great question. Thanks. So the question was, what are we doing to condition the urchins prior to release to get them ready to deal with life on the reef? Right? Is that the question? Yeah. So not much. We do something. So yeah, we have done some behavioral studies, right? So the urchins are actually nocturnal foragers, and so they go out at night and then they do their feeding and then they hide during the day, and that's probably a strategy to avoid being eaten by fish and other predators during the day. And so we were curious to know if the cultured urchins would behave similarly to wild urchins. Because if they didn't, and they imagine they were foraging during the day, then they would get eaten really easily. And thankfully, they did behave in the same way. And we think it's because they had some period of conditioning in a greenhouse aquarium that had a bunch of natural rock and structure in it, and also a natural photo period, so that they could kind of they're not just in an empty tank with basically nothing for them to hide within. If that answers your question, yeah.
What about sea swells and stuff? How have they coped? Because they're not put in a tank which actually oscillate or replicates sea movement, are they?
Yeah. So the question is, what about like ocean currents and swells, and are the urchins sort of conditioned to deal with those in an aquarium? Yeah. So not really. I mean, there's a bunch of circulation that's really important to have in an aquarium, but we don't. It's hard to replicate like an ocean swell in an aquarium, and so that's a really good question, and maybe something that we need to look into. But I think most when they're on the reef and there's a big swell, they could hide in some structure, or they have ways to maybe they'll go into deeper water or something. And so hopefully, they'll know to do that. But if it's too rough, then yeah, it could be sort of a mortality event for them. Yeah, that's a great question.
I mean, as we know, the sea levels are rising,
Mm-hmm.
and therefore they are moving northwards.
Mhm.
Has anybody, or is it too early yet, to have tried to establish a coral reef in a slightly out of the normal area?
Yeah, that's also a really good thought. And so there's probable range expansion that will happen, and it depends. For instance, in Florida, the currents on the eastern part of Florida are such that it goes from really historically good habitat to probably bad habitat really quickly. And so we don't have a lot of room to do that type of work in Florida. But I think people are probably looking at that, and because it's not just the habitat isn't simply a question of temperature. There's other parameters that lead to sort of a coral reef establishing in certain areas.
But surely temperature is actually going to move other creatures, fish, etc., because they are not able to cope with the rise in sea temperature where they are now,
Right.
and will perhaps the whole lot will move together.
Perhaps, and that would be really nice to see. Yeah, I think it's a really good question. I don't think I can speak too much to that right now, but it's a really, really good thought. And yeah, maybe we'll see more of attempting to do some of the restoration work slightly outside of their current thermal profile. Yeah, or range. Yeah, that's a really good question. Yeah, thanks. Yes? Oh.
Thank you, everybody. Can I have a show of hands of who needs a taxi, who is not staying for dinner? 1, 2, 3, 4, 5. Okay, 6. Coochie is right here at the lower driveway. He's ready to whisk you off to your next plan. Thank you for coming, and we'll hope to see you on Tuesday as well. For people having dinner, obviously, you can head up toward the bar area. We're going to help them get ready chair-wise. We'll move them, or you could just put them over to the side. But thank you, everyone, for coming.
See you next time.